Welcome back to the lab. In our last episode, we learned how to read the code of life with DNA sequencing. But before you can sequence DNA, you have to prepare it. Today, we're revealing the essential, often overlooked first step: DNA library preparation. We'll show you how to turn your raw DNA into a format that a sequencing machine can actually read. And we're back. Think of DNA library preparation as the crucial link between your DNA sample and the sequencing machine. It's a critical step that converts your fragmented DNA into a format that is compatible with sequencing platforms. This process has evolved significantly to address challenges like degraded DNA and low-input samples. The main goal is to create a library of DNA fragments, all with special adapters attached to their ends. These adapters allow the fragments to bind to the sequencing machine and be amplified. The basic workflow involves a few key steps. 1. Fragmentation. We start by breaking the DNA into smaller, manageable pieces. 2. End repair and ligation. We repair the ends of the fragments and then glue on the adapters. 3. Amplification. We use PCR to make many copies of these newly created library molecules. If you need a refresher on how PCR works, be sure to check out our previous episode on that. These carefully prepared libraries are what get loaded onto a sequencer to begin the decoding process we discussed in our DNA sequencing episode. Now let's get into the two main types of library preparation methods you'll encounter on the bench. 1. Double-stranded DNA, or dsDNA library preparation. This is the standard, most common approach, and it's perfect for high-quality DNA samples. It involves the steps we just mentioned: fragmentation, end repair, adapter ligation, and PCR amplification. While this method is widely used, newer protocols have introduced enzymatic fragmentation, which improves yield and reduces artifacts, especially with challenging samples like FFPE tissues. 2. Single-stranded DNA, ssDNA library preparation. This method is a game-changer for difficult samples, like degraded or ancient DNA. Instead of working with double-stranded fragments, this method converts each individual strand of DNA into a library molecule, which dramatically increases the recovery of genetic information from highly fragmented samples. Key steps in ssDNA library prep include the ligation of adapters to the single-stranded DNA and a strand synthesis step. Modern one-tube and rapid ligation-based methods like srSLY reduce processing time and cost, making these approaches perfect for high-throughput and sensitive workflows. Because this is such a critical step, troubleshooting is essential to get high-quality results. Here are some key issues and solutions. Problem 1. Low-input DNA. Solution. For samples with very little DNA, use dedicated single-stranded library preparation methods. These are designed to increase the recovery of genetic material from highly fragmented or damaged samples. Problem 2. Degraded DNA. Solution. Degraded DNA, like what you'd find in ancient samples, often works best with ssDNA library prep, which is specifically designed to handle highly fragmented templates. Problem 3. Poor yield or low-quality reads. Solution. Low-quality library preparation can lead to low-quality sequencing data. Ensure your sample is of high quality and consider using enzymatic fragmentation protocols, which improve yield and reduce artifacts. You should also carefully optimize the number of PCR cycles to avoid amplification bias. By paying close attention to these details, you can ensure a successful library preparation and reliable sequencing results. So there you have it. DNA library preparation isn't just a simple step. It's the critical foundation for every successful sequencing experiment. It's the process that turns raw, complex DNA into a perfectly structured library ready to be decoded. Without it, the incredible world of DNA sequencing would be impossible. If you're fascinated by the tools that unlock the secrets of genetics, hit that subscribe button and ring the notification bell. Our How to Biotech Lab playlist is your go-to resource for understanding all of these techniques. Join us next time as we explore another exciting frontier in the world of biotechnology.